Good afternoon, students. Today we are going to discuss the next lecture of molecular biotechnology, and that is all about molecular markers. These molecular markers are divided into two categories. The very first is that is no more based on the principle of PCR, and the second category that we will be talking about is the category that is based on the PCR. That means the name itself is indicating non-PCR means the molecular markers which are no more following the principle of PCR, that is the polymerase chain reaction, and the other that follow the principle of PCR. So these four contents we are going to discuss today. Very first we will be discussing is introduction. Like before I actually come to the PCR and non-PCR based markers, one must be familiar with molecular markers. That all will be discussed in the terms of introduction. Then we will cover non-PCR marker. And when we talk for the non-PCR marker, then we have only one category under it, and that itself is called as restriction fragment length polymorphism. And then after the non-PCR, we will talk about PCR based markers. That itself is involving three kinds of molecular markers. And thereafter, we need to talk about variants of these markers, right? So very first I come to the introduction, what actually is meant by the molecular marker. So what is molecular marker? Molecular marker is the one that is identified as genetic marker. Now genetic marker means you can understand something. Even in your say daily life, daily basis of life, you used to have a marker with you, that is a pen. Two kinds of marker you must be using. One is permanent and another used to be the temporary marker, right? Even when we talk for the living organism and when we are talking at the level of molecules, then also we used to have markers. But because molecules are all, when we talk about the molecules, we all talk about the genetic content. That means this molecular marker is identified as a genetic marker. And when we say genetic marker, that means where comes the term genetic? There comes DNA. So any guesses what can be the genetic marker? So genetic marker is a DNA sequence. It is a DNA sequence. If we don't call it as a DNA sequence, then it can be said to be a gene sequence. But this sequence is placed on a particular location on a chromosome. This is chromosome. Now, I will be asking you at the end, like, what is the difference between DNA and chromosome? Because we said molecular marker is a DNA sequence. DNA sequence or say gene sequence, right? And this DNA is located onto a chromosome at a specific position. Say it is located here at a specific position. Because these are called as markers, because see, it is very, it must be very clear to you all. Molecular marker means the marker. It is marker. When you used to read, say, particular lines, you used to read, what you used to do? You highlight certain lines with this color. That is called as a marker. And that means something, it is very important, right? In the similar case, when we are to define the molecular marker, what we do? What we do when we are to define the molecular marker, we say molecular marker is a sequence, is a DNA sequence. That means DNA sequence, when we say, then we are all talking about the sequence of adenine, thymine, guanine, cytosine. This is the DNA sequence. When we talk for this recognized DNA sequence, which is placed at a fixed position onto the chromosome, then we call it as a genetic marker, right? Because it is always available at a fixed position, then it is very easy to say, like because its position is fixed onto the chromosome, so it can be utilized as an identification tool. For example, I say this is the one paragraph that is given to you. I say what is the important line, and I told you that important line will be the one that will be marked with this that will be underlined with this color. So this is a recognized line, right? And it will be utilized to follow what words or which sentence is very important in on this slide. In the similar way, this is the DNA sequence, and the DNA sequence that is present within a recognized location onto a chromosome. It is used as an identification tool. That means when that is we will always be sure, like if any, suppose we don't know what marker is this, but if we know that the location of the marker is so and so onto the chromosome, then we can easily say this is a kind of so marker, right? One more example I let you know that will help you to know more about the molecular marker is, suppose we have a pool, and in the pool we have unknown DNA. This is the pool, and in the pool we have unknown DNA. These are the unknown DNA, right? But if at any place is the presence of this molecular marker, this is a molecular marker that was present here, the sequence of which is A, T, G, C. If these are the unknown molecular markers, then these, if there is anywhere present, is present a molecular marker that is having the same sequence as was present here, then we, out of this unknown pool of DNA sequences, we can very easily recognize this one, right? Because this sequence is known, we will say so and so sequence used to be present in so and so DNA of so and so organism, right? So when we talk for the unknown DNA, when we have a number of unknown DNA, then in that particular case, molecular markers are only the one that helps in the identification of a particular sequence of DNA that is present at a particular location. That means if we have this sequence of the DNA, say this sequence is adenine, thymine, guanine, cytosine. Say we have this DNA sequence, and here will be the complementary, right? But say if we have another sequence A, G, T, and C, and here will be the complementary sequences. So how can we go for defining the molecular markers? Molecular markers are the one which reveal the variation at DNA level. That means whatever changes that are taking place within the DNA level, within the DNA at the molecular level. Molecular markers means the markers which are revealing the variations, which are revealing the changes, which are letting you know that what change is taking place. See, this is the change that is taking place, and this can be, this is at DNA level, and we can say it is at molecular level. So the markers which reveal, that means which explains the variations at DNA level, they are referred to be the molecular markers, right? So this is all about the molecular markers. Because molecular marker is revealing the variation at the DNA sequences, and DNA is a genetic content, so they are also identified as genetic marker. When we talk about that sequence, then molecular marker is a DNA or gene sequence that is present at a, this is very important. That means its location is always recognized onto a chromosome, because its location is not changing. Say if I say these two DNA are of same kind, and say here should be, this is the recognized location of the molecular marker onto these two chromosomes. But if I say this molecular marker is present here, this is not more possible. because position of a particular molecular marker is always fixed onto the DNA. And this is only the reason because its position always remain fixed within a specific kind of DNA. So this is only the reason it is always called, it is always used as an tool for the identification of unknown DNA, right? Now, very next question that must be coming into your mind is about its application. Application is, very first application is it plays a crucial role in gene mapping. Gene mapping means what? When we say mapping, when we say mapping of India, then in the mapping of India, we come to know which country, which state is located where, and India is connected to what all other countries. That is all about mapping, right? But here we need to talk about gene mapping. And when we call for the application of molecular markers, then the very first importance of molecular marker is in terms of gene mapping. That means it plays a crucial role, and how it is playing a crucial role by identifying, say, if this is the location of molecular marker, and say the genes near to it are G, and G, then C, then A, then T. This is the sequence, right? And this is the location of molecular marker. Then how it helps in the gene mapping? This molecular marker is helping in the gene mapping by identifying the position of the linked genes. Linked genes means the genes which are linked to each other, and when there takes a phenomenon the phenomenon that is said to be inheritance, and when these genes are transferred together to the next progeny, they are linked and they are transferred. That means they are inherited together to the next progeny. In the gene mapping, it plays an important role in identifying the position of these linked genes. That means where these linked genes are present, that in turn can be analyzed by the molecular marker. Secondly, if there is any change in the sequence of the DNA, it can be very easily identified or detected by the molecular marker. And due to the change in the variation, if there is the occurrence of any genetic disorder, that in turn can also be determined by molecular marker. Third, it helps in the determination of different character in the DNA sequence. Like this is a DNA sequence, and on the basis of the different characters that are present in the DNA sequence, it is helping in the identification of individuals among the population or species. Like on the basis of these characters which are present in the DNA sequence, we come to know whether the species are homozygous or whether the species are heterozygous or not. So this in turn can also be identified. This can also be identified by using the molecular markers. Now, once you came to know about the molecular marker, and once it is very advantages of the molecular markers are very clear to you, the very next question that must be coming into your mind is like, what is to be used as a marker? This much is clear to you that molecular marker is something that is having a particular sequence and it is located at a particular location onto the chromosome, and it is helping in the analysis of helping in the analysis of DNA that may be present in the pool containing unknown DNA, right? This is clear to you all. But what is to be utilized as a molecular marker? So, because when we talk about the variations, we always say, and don't forget, when we talk for the variations, even in the agriculture, variations are always taking place at the genetic level, say at the gene level. And when we say gene, then it is very clear we are talking about the DNA. Because large number of variations, large number of changes, they are taking place at the DNA sequence level. These variations can be used as molecular markers. These variations can be used as molecular markers for the evaluation of variability that is taking place in terms of phenotype. Again, I repeat, what is to be used as a marker? Because you said marker is having a specific gene sequence. A specific gene sequence is okay, it is clear to us. Specific gene sequence means sequence is defined, say it is A, T, G, C. But when variations are taking place within this sequence, then those variations will also be known. Say the variations are A, G, C, T, or say the variations are something else. So these variations only are the basis of molecular marker, and these variations can only be utilized for the identification of molecular, say for the identification of phenotype. If you further wanted to understand the concept of molecular marker, we need to know about its features. So the features are actually based on the techniques which are used for the detection. Like very first, it must be polymorphic. Polymorphic means it must be having a number of variations so that the diversity could be measured. Because if it is always having a fixed sequence, then we cannot come to know about the variations. So it must be polymorphic. That means polymorphic means it must be including maximum of variations, because more will be the variations, more will be the diversity measured. Secondly, it should be distributed throughout the genome. Obviously, if it is not distributed throughout the genome, then if it is not distributed throughout the genome, like it must be distributed throughout the genome. Because if it is not distributed throughout the genome, then we can only analyze the portion where it is, where we can only analyze the portion of the genome where it is actually located. And it will be difficult to analyze the other portions of the genome where it is no more located. So this is the reason we say it should be distributed throughout the genome. But the important parameter is molecular markers, they should be easily and fast detected. It should not be like you are given a pool of unknown DNA, and within the unknown DNA, you yourself don't know what actually is the sequence of say molecular marker. And it is taking number of years for you to recognize very first the sequence of the molecular marker. And by the time you recognize the molecular marker sequence, it is very late and you are taking too long time for the identification of DNA that are present in this unknown pool, right? So it should be one parameter that allows the any variation to act as a marker is it should be easily and fast detected. And because variations can result into the homozygosity or heterozygosity, so it must be able to distinguish homozygotes and heterozygotes. So these are the certain features that one must possess for acting as a molecular marker. Now I will talk about the classification of molecular markers. Very first classification includes the category that is no more based on the PCR, and they are also called as hybridization base markers. And the second classification is the one where markers are based on PCR technology, PCR principle, and they are said to be PCR based markers. Now these PCR based, this hybridization is the one that is involving restriction fragment length polymorphism. and these two are the one that is based or that comes under the category of PCR based markers. The use of restriction fragment length polymorphism is a non-PCR based approach, and the other two, these are the PCR based approach, right? Now we will talk for non-PCR based markers. And the example in the non-PCR based marker is random fragment length polymorphism. Random fragment length polymorphism, and the full form for it is Rflp and S. R stands for random, F stands for fragment, L stands for length, and the P stands for polymorphism, and this S is denoted by a small s. So this is non-PCR based molecular marker, or you can say if it is non-PCR based, the principle behind it is hybridization. Okay. Now very first is this Rflp. Of course, these are the genetic markers. See, before I actually talk for this genetic marker, I would like to tell you, say we have a DNA, and the length of the DNA is defined. Defined means length is fixed. Say if this length is changed, why the length is changed? Because certain restriction enzymes, that is restriction endonucleases, they acted upon the DNA and they digested the DNA and result is like we had a DNA that is of shorter length as comparative to the original DNA length. Now, these are the markers which are resulting from the change in the length of DNA fragment. Markers usually used to be present here. Say this is the marker, this is the marker, this is the marker. Now the marker which are resulting from the change in the length, three I have represented. This is one and these are the two. So markers which are resulting from the variation, that means resulting from the change in the length of defined DNA fragment, those DNA fragments which are produced as a result of digestion of the DNA sample, they are said to be molecular markers, or they are said to be genetic markers. And which kind of genetic markers? Rflps. See, name itself is indicating random fragment length polymorphism, because when it will be acted by a number of restriction enzyme, then fragments of different lengths will be produced. Fragments of different lengths will be produced. Say this is, if this is A, this is B, C, D, then the fragments of different length A, B, C, then say this is D. When the fragments of different lengths, but all these fragments are having one marker located onto it. So these are said to be random fragment length polymorphism, because randomly they are being digested by the restriction endonucleases. So this is the reason the fragments are produced randomly. Randomly means they are produced of different sizes, and these different sizes, the DNA fragments which are produced as a result of digestion by restriction endonucleases, they are said to be genetic markers. This is clear to all. So this is random fragment length polymorphism. More it will be clarified. So when we talk for the features of random fragment length polymorphism, then one organism can be differentiated by the analysis of patterns which are derived from the cleavage of the DNA. Because here different length fragments are produced as a result of action of the enzyme called as restriction endonuclease. So this technique is mainly based on special enzyme. Again I repeat, in the Rflp restriction enzyme, they cut the DNA, and the DNA that are cut, they can be resolved. Resolved means they can be placed in one sequence, like which the smaller one be here, the larger one will be this. They can be resolved. Resolved means they can be placed in this sequence by a procedure that is said to be gel electrophoresis. And you know, in gel electrophoresis, when we actually perform the southern blotting, then the gel is later on placed onto the cellulose membrane. So how resolution of these different fragments which are placed, which are produced as a result of action of restriction endonuclease, how they can be resolved? So their resolution is very easy. This gel that is containing different segments of different sizes, they are placed onto the gel electrophoresis. So you know, when we are placing on the gel electrophoresis and switching on the electric current, then under the action of electric field, the smaller one will move faster comparative to the larger ones, and they all will be visualized in terms of bands, right? Now which band is which one? This in turn can be visualized by hybridization with probe. Probe is the again known sequence. Different size fragments are produced, and these different fragments are said to be polymorphism, that is the variation, and these variation in turn are used to distinguish the plant species, genotypes, etc. We continue with the random fragment length polymorphism, and if we talk about its history, then we'll come to know this technology was very first time employed. It was utilized for the detection of polymorphism, that means the variations based on the DNA sequence differences. This is now very clear to you all. When we say a random fragment length polymorphism, then we talk for the DNA sequences differences only. This is mainly based on restriction enzyme site, and outline is given in the next slide. What about procedure? This is the procedure that involves the very first is that involves the isolation of genomic DNA. After the isolation of the genomic DNA, this DNA is digested by the restriction endonuclease enzyme, that in turn produces DNA pieces of various length. Then these DNA fragments are to be resolved onto the gel electrophoresis. So here is carried out their electrophoresis. And after the electrophoresis, they are very important step. Here, after the electrophoresis, once the DNA are to be transferred onto the filter paper, nitrocellulose filter, that is southern blotting, and then these pieces are incubated with labeled probes. So see, this is the procedure that is involving very first step isolation. After the isolation, digestion of the DNA. After the digestion, separation of different length DNA fragments by the procedure say electrophoresis, and finally hybridization, right? So see, this is what we have discussed here. Isolation, this is digestion. After digestion, their resolution, and after resolution, hybridization. So this is an outline of restriction fragment length polymorphism. Here we are no more multiplying the number of the DNA. We are just restricting them. This is also considered as how amplified products are to be separated. Again, we had an individual. From the individual, we isolated the DNA. DNA is basically double-stranded DNA. Then this double-stranded DNA is denatured, that is converted to single-stranded DNA. Primer is annealed to it, and complementary strand is synthesized. These are the steps of RFPD analysis by using PCR. That is random. This is not the procedure that is to be discussed here. This is actually RFPD, that is PCR based marker. I have attached this PK here so that you could differentiate among the two. This is restriction fragment length polymorphism, where what is done? Digestion. After digestion, separation, then transfer onto the filter, and then hybridization. But if you could see here what is done? Isolation. After isolation, we used to have double-stranded DNA. In this non-PCR, this double-stranded DNA was being divided into number of the pieces. It was being, say, separated into the number of the fragments. But here no fragmentation is carried out. Here, if you could visualize, then double-stranded DNA is denatured. And this is what the principle of PCR, right? This is what the principle of PCR, where I already said we are following this denaturation, then annealing, and then say extension. This is what? This is PCR. And here, since no PCR step is followed out, here we are isolating. After isolation, it is fragmented into different pieces, and then hybridization is carried out. That means we are no more denaturing the DNA. We are no more attaching the annealing the primer to it, and no more we are carrying the extension. So this procedure is said to be non-PCR based, and very famous example of it is Rflp, that is restriction fragment length polymorphism, right? So here what is done? If you could see this flow chart, again I'm explaining the procedure only. If we see this flow chart, see we had plant tissue or say animal tissue, depending upon your requirement. We isolated the DNA. The DNA that is isolated is said to be genomic DNA. See here, when genomic DNA of many individuals of one species are separately cleaved by this is now cleavage by restriction enzyme, and then they are passed on to the electrophoresis. That means separated onto the gross gel. Then when passed on to the electrophoresis and when gel is placed onto the nitrocellulose membrane, we call it as a southern blotting. Onto the southern blotting, now we want to recognize the DNA. So the DNA, they are probed, hybridized with radio-labeled DNA. So the sequence of this radio-labeled DNA is clear to us. Wherever there occurs attachment of this radio-labeled DNA, because we knew the sequence of this DNA, so the complementary sequence, wherever it goes for binding, that complementary sequence can be analyzed. And as a result, there arises variation in hybridization pattern. Like some will hybridize at the first location, some will hybridize at the second location, some will hybridize at the third location. So variations will arise due to the hybridization patterns of digested DNA. These variations, that means the differences in the sequence between two individuals, very important. These differences are very unique, common, and repeated DNA sequences which are found in several individuals. These are said to be random fragment length polymorphism. Random means they may be distributed anywhere within the DNA. Fragment means the fragment that is produced as a result of digestion of DNA by restriction endonuclease. because it will be digesting the DNA at different locations, so different lengths will be produced, and polymorphism means a variation. So after digestion with one specific enzyme, if we talk for only one specific enzyme, then variations will be obtained with that enzyme is only. That means I mean to say for example, if this is the DNA and we are talking for only one restriction enzyme, otherwise we said number of restriction enzyme and they are producing the different length, right? But here once the DNA is digested with a specific enzyme only, then in that case variation in this one DNA fragment with this enzyme is referred to as 1 Rflp. If I say this is a second restriction enzyme, then due to this restriction enzyme, whatever variation will be produced in this second DNA, it will be called as second Rflp with concern to this restriction endonuclease. So this is about restriction fragment length polymorphism, where as a result of restriction by restriction endonuclease enzyme, different length DNA are produced, and all those DNA they vary in the genetic variation, and this is said to be random fragment length polymorphism. Name at itself is indicating random fragment length polymorphism, right? This can be better understood by an example. Say the example is imaginary example where we have three plants. Here we have three plants A, B, C. Out of these three plants, this is the original plant, A is the original plant, and B and C are the mutant one. What are the features of original plant A? That is, these are the restriction sites, okay? And this is the restriction map. Like the restriction map is having is consisting of sites that is of 12 kb fragment that involves a restriction fragment of 12 kb, 8 kb, 6 and 3, right? This is original plant that I have already told you, but how you will come to know these two are not the original and these are the mutant one? Because for original plant we know restriction map is this one, that is altogether including of 4 lengths, right? But within the B there had been the appearance of this one extra band and this extra band. That means when we talk for the mutant B, then within the B there appeared extra fragments of 7 and 5 kb. That means this is 7 kb and this is 5 kb. But when we talk for the C, then in spite of 7 and 5, there is one more additional. There is no 6. Here is within the B there is appearance of the additional fragments that are said to be 7 and 7 fragment new restriction site appeared in the 12 kb. This is together 12 kb fragment, and that 12 kb fragment is involving additional fragments of 7 and 5 kb. Okay, but when we talk for the mutant C, then in the mutant C, this 6 kb is lost. This is lost, and we have the appearance of 10 kb. So this is the restriction map. I mean to say these are the restriction sites which are produced in this 12 kb size fragment. B is said to be mutant because here 12 kb fragment is lost. Even here it is lost, and the additional appearance of 7 and 5 is taking place. And in this case, fragment of 12 and 6 is lost, and additional appearance of 10 and 5 is taking place. So this is non-sequence, and these are the unknown sequences. So this is the reason this is said to be a restriction fragment length polymorphism. That means this is the restriction fragments produced, and this is the whole length, and the variation that is involved in this whole length is said to be restriction fragment length variation, or often it is called as restriction fragment length polymorphism, right? So the next is conclusion. So what could you conclude from restriction fragment length polymorphism? That means these are the codominant markers. Codominant because they are capable of identifying heterozygotes from the homozygotes at the population level or at the individual level. Then it identifies a genotype in any tissue at any developmental stage. And they are also detected by using sequences of the DNA, and as a result, Rflp map can be constructed. Here is the preparation of Rflp. There are 3 steps we will talk for that is involved in the Rflp. Very first is how we are to go for the preparation of genomic DNA probes. Genomic DNA probes means probes is a specialized sequence. So there are many DNA or RNA probes. DNA or RNA. DNA means double stranded. That means there will be the presence of T, and here in a RNA there will be the presence of U. So these probes are used. These are used to recognize the complementary sequence, because if their sequences are known, wherever they are going to hybridize, other complementary sequence can be very easily recognized. These are preferred over other probes. These are preferred over other probes, and for the construction of Rflp maps. These are the steps that are, this is very clear to you all till now. Very first we are extracting the genomic DNA, then we are digesting it, then we are transferring it onto the gel, where all are getting resoluted on the basis of their size. Then we are isolating the desired and joining. So these are the steps. Very first is the isolation, then digestion. After digestion, the digested DNA, they are separated on the basis of their size. Then we will isolate the one that is required to us, that means of a specific size. We will join it into the vector. We will transform the recombinant vector, and in this way cDNA probes are prepared when we keep on using step 2 to step 4 are repeated again and again. Second, these are used for the detection of Rflp. In the detection of Rflp, this also I have told you, first of all you need to collect the sample, then it will be fragmented, then it will be electrophosed, bloated, that means southern blotting, where it is transferred onto the nitrocellulose, and fragments can be observed by hybridizing with the probe and seeing it under the autoradiography, right? Very important. Those fragments which will get hybridized are said to be restriction fragment length polymorphism. Then uses, these are the first class of genomic markers that allows the construction of highly saturated linkage map, and there are the 3 important ways by which it can be used. That means in the indirect selection, tagging the monogenic trait, and indirect selection. We need not to go under these say uses in details, but we must know these 3 are the main uses of Rflp. With this, I come to the completion of non-PCR base marker, and under which we only discussed one and only one molecular marker, that is called as restriction fragment length polymorphism, right? Now we are to discuss the PCR base markers. These are the PCR base markers. Very first PCR base marker is random amplified polymorphic DNA or Rflp. The outline of which I told you while letting you know the study flow chart of known and PCR, right? So it is very clear because they are coming under the PCR base markers. So they are based on PCR amplification. This is the outline where DNA is isolated, denatured, annealed, extension, right? And here short oligonucleotide primers can be selected and used for the amplification. Whatever products are amplified, they are separated on electrophoresis and then these are identified. This is all about the history of a random amplified polymorphic DNA. For the very first time, it was developed by scientist named William and his co-workers in the year 1991. Of course, it is under the PCR technology, so it is based on the PCR technique. It is widely utilized in the molecular biology and very important. Its function is to amplify specific segment of DNA. Its function is to amplify specific segment of DNA. But if see, because it is used to amplify specific segment of DNA. Say if you have information about these are the DNAs, set of the DNA. Say these are the set of DNA. If you have some information about these set of DNA fragments which are randomly distributed throughout the genome. These fragments are randomly distributed. Say this is one fragment, this is second fragment, this is third fragment, and here is the fourth fragment. If you have a set of the DNA that are randomly distributed throughout the genome, say if this is distributed here, then it may also be distributed here. That is, it is randomly distributed. There is no sequence of distribution like this is the one, so it will be on the one number only. It is on the two, then it is on the two number only. No, there is no this can be 3 and 4. That means no sequencingly arrangement. I mean to say, if you have information of set of the DNA, then you can synthesize. If you have information of this DNA, normally what we used to have, we don't used to have information about this DNA. We used to have information of DNA or RNA probe. We used to have information of DNA or RNA probe, the sequence of which is known, and wherever it hybridized, that complementary sequence is analyzed in that on that basis. But here the situation is opposite. Here we are having the information of these DNA fragments, and since we have this information of the DNA fragments, that means its sequence is very much known to us. Then on the basis of this sequence, what one can do, we can synthesize the primer. Normally, we used to have information of the primer and we used to detect the sequence of the DNA fragment. But here the condition is opposite. We have the sequence of the DNA, and on getting the sequence of the DNA, we can synthesize the primer, and then this primer can be attached to the both the ends of this particular DNA segment, and this segment can be amplified by a process that is PCR. This is clear to all. So this is RAPD. That means if you are within the RAPD, if you remember within the restriction fragment length polymorphism, there were variation in the sequences, and those variations were itself said to be restriction fragment length polymorphism. But here we have a random amplified. That means these are the sequences that are randomly distributed through the genome, and since the sequence of these fragments are known, opposite to it, primer can be synthesized and primer can be attached to these both ends, and DNA can be amplified. Because the fragments are distributed randomly, and we are randomly amplifying these fragments. So this is only the reason it is said to be a randomly amplified polymorphic DNA. And the primers which we are synthesizing, they can have the bases into it 10 moles. DNA amplification, this is very important, as I have told you in the previous slide also. DNA amplification will only take place if it is flanked by a part of 10 base pair, right? And as a result, the random sample of DNA marker is obtained. So this is called random amplified polymorphic DNA. But this must be noticed, like if for one genomic DNA there are different RAPD that are existing, and for the genomic DNA of other individuals, RAPD are different. So specific DNA segment that shows the DNA polymorphism, so this can also be utilized as a genetic marker. These are also available in the market. If you don't want to go for the synthesis of the primers, they are available in the market. What is advantages? Here we know, because you yourself can synthesize the probes, here no specific species specific probes are required. Secondly, data can be very easily collected. In comparative to RAPD, we are getting the data 5 times more quickly than the time that was being taken in a restriction fragment length polymorphism. Only a small amount of DNA is required because DNA is randomly being amplified. So you need not to have amplified DNA in the initiation only. DNA in the initiation is very lesser, and that in turn can be amplified as a result of RAPD. This is also the very important difference. Earlier hybridization was taking place. Here it no more requires a hybridization. So these were the advantages. These are the applications. You can go for the preparation of genetic map, and genetic maps with the help of RAPD has already been developed in terms of arbediopsis and helianthus and pines. You can map the genetic traits. That means you can also come to know resistant traits against pathogen, etc. It is also involved in fingerprinting of the individual organism, and certain genetic markers, they may be tagged at specific regions also. So this was all about restriction. This is all about a random amplified polymorphic DNA. Now we come to the another category of PCR based molecular marker, and this is said to be microsatellite, and it is often another name of it is simple sequence repeat, that is in short abbreviated by SSRS. It was discovered by Litton Luty in the year 1989, and here base pair are only 1 to 6 base pair long. Here different length motif may be present, like only single base is present. These are 1 to 6 base pair long, no? So variation is there, like in turn one base is present, or either 2 are present, or 3, 4. So different length motifs may be present. They are also randomly present throughout the genome. And because it is randomly present, but it is present in very abundance. So these are used as a DNA marker for mapping. If a very high level of variation is taking place at allelic means alleles are the 2 alternative forms of the genes, then this in turn can be demonstrated using PCR. And these are the most common ones that are utilized. They are easily to use, and they can be cloned and characterized later on. All the time they show the variations. Variations are always of a nature that is stability. That means they are always producing stable polymorphism. These are the ideal markers which are considered for high resolution generation of molecular maps. So this is a bit of about simple sequence repeat, and the last is amplified fragment length polymorphism. Till now, you studied two kinds of molecular markers, one that is based on hybridization and another that is based on PCR, amplification. But AFLP, that means amplified fragment length polymorphism, is a combination of both. And you know, it is useful in detecting the variation throughout the genome. It is very sensitive. Whatever kind of variations are present throughout the genome, because here see, we are utilizing both non-PCR and PCR based markers. So even if any variation is left as a result of Rflp, that in turn will be covered by RAPD, and if any variation is left by RAPD, this will be covered by. That means this is a very sensitive method that will let you know about a small to small variation that is existing within the genome. So this is the reason it is very superior to RAPD also. And because variations can be in terms of difference or in the terms of same genetic content, so it is able to distinguish hetero to homozygote. It is applied universally just because of the reason its reproducibility is taking place at a very high level. But the basis of it is PCR amplification only. But very important, because here is the combination of the two techniques. That this is involving PCR amplification, and this involves a production of random fragments. So here amplification of restriction fragments that are produced will be taking place. That means amplification of restriction fragments will take place. It is a novel technique, and it is based on amplifying certain restriction fragments, right? That I have told you in this step only, this one only. This is the basis of this procedure. Principle is again very clear, because here what is to be done? Here both the techniques are combined together. So first of all, fragments will be produced, and later on those fragments will be amplified by PCR. That means here digestion and PCR both will work together. It is a new technique that is utilized in DNA fingerprinting, but very important. It requires a very stringent, that means specific conditions for the primer annealing. Like you have read, no? When we say primer annealing in terms of PCR, then we say PCR, within the PCR at the step of primer annealing, temperature is to be lowered. Like in the denaturation, we increases the temperature to 92 degrees Celsius. But at that 92 degrees Celsius, there won't be any attachment of primer. So primer is to be attached by lowering the temperature to 74 or near about. So it requires stringent, that means very specific conditions. Otherwise, it won't be working, this process won't be working out. It is also used in mapping of genome, but since it is involving hybridization, it is involving amplification, so it is a expensive technique. This is the principle. Because AFLP, again I repeat, AFLP, amplified fragment length polymorphism, since it is the combination of two, that is RAPD and Rflp. Very first step here is what? Isolation of the DNA, and later on its restriction and production of fragments. These fragments here will be joined together, and then they will be amplified. And the third step is the amplified products will be later on transferred to the gel. Gel is a kind of agarose gel and it will be subjected to electrophoresis. In the electrophoresis, these DNA will get separated on the basis of their size. And these in turn can be, because here this attachment of Rflp, that is dependent upon hybridization by probe. It is based on hybridization principle. So these hybridized fragments will be identified by autoradiography. These will be identified by autoradiography, where, because hybridization is with the radio labeled isotope, yesterday also I told you, which fluoresce under the UV light, and wherever there will be the emission of the color, we will come to know this color is due to the presence of attachment of is due to the attachment of probe, and that particular sequence can be detected because we know the sequence of the probe. In case these radio labeled nucleotides are not available, we can go for the use of fluorescent dye or silver staining methodology also, right? So this is all about amplified fragment length polymorphism. You must be thinking, no, why we need to understand so about molecular markers when we have conventional breeding with us. So here we'll clarify more of your concept, like why we need to go for the molecular breeding. You know this conventional breeding. What is conventional breeding? I need not to tell you. You have already read in the 6th semester number of the breeding technologies that were used for the handling of segregating generation. Either it is mass, pedigree, say it is backcross, any, right? But even you must be remembering these conventional breedings were taking many years to accomplish, many years to complete. But here, with the help of molecular markers, you could easily recognize the variations at the DNA level. You need not to wait for the say 10 to 12 years, of say for the 12 to 15 years. It is very speedy method of identifying the variations at the genomic level, and later on those variations may be utilized for any purpose. You may utilize it for say utilization maybe for amplification, utilization maybe for the identification, any of your purpose, right? But basically we are amplifying for revealing the genetic variations only, right? So here breeding is all in this breeding you are only based on the morphological features, that means the phenotypical features. But here recognition very important, but here the recognition is being revealed at the differences that are taking place at the DNA level. And you know, all is the game of DNA only. When we talk for the molecular biology, when you are talking for the agriculture, when you're talking for the breeding, all the concept is revolving around the genetic content only. But here these expressions are also produced as a result of genetic content only. But these expressions are these expressions, although these are the results of genetic content only, but these are the phenotypic features, and it is not always even if you remember phenotypic features, I told you they are affected by the interaction that takes place due to the environment also. So we need not to depend upon these morphological features, right? Although recognizing any plant on the basis of morphology is very easy, but this is not an easy task. This is a very sophisticated laboratory technology, and that is to be utilized very carefully, right? This I have already told you. Phenotypic features, they have high effect of environment, but genotype is no more having any effect of the environment. Accuracy is medium to high, but in this case, accuracy is always and always at a higher level. Here, see, when you go for the conventional breeding, you need to occupy at least 10 to 15 years. And even you don't know that at the end you will be receiving the results that you will be getting will be the accurate or not. And even if you remember, number of trials are to be conducted in between these 10 to 15 years. Say these trials may be conducted at say 7th year, may be conducted in the 8th year, or you may be rapidly conducting these preliminary yield trials and multi-locational trials again and again. But here the time period is very low, that is, it is only all about 3 to 5 years, and we need not to go for the preliminary trials and all. Of course, it is taking time consuming, it is time consuming, but the cost involved is low to medium. But it is very sophisticated laboratory techniques, and whenever we talk for the latest technology, of course, we are to use certain equipments, and you know, PCR is only the instrument that is coming in, I don't know how many labs. Individual equipment PCR I'm talking about. You just Google the prices of the PCR and you will come to know PCR is a very costly instrument, right? Here, here hazardous effects are only present in case any mutation breeding takes place. But in this case, in this case, this technique is here because you remember I said when we are using the non-PCR ones, then we are focused on the hybridization. Hybridization means we are having the non-sequence of probe that will be binding to a complementary region, right? But here when we say, but that probe is further attached to the radio labeled isotope, and that radio labeled isotope will emit the light under the UV exposure. So this also is producing certain health hazards when you are using the radio labeled probe. Fine. So here effect of gene interaction is very high. Here gene to gene effect is no more possible. No effect is taking place. Here we can, this is quantitative trait loci. Here mapping is not possible because you are no more familiar with the genomic organization. Then how you will go for the mapping? And here mapping is very easy to do because as I gave you imaginary example, and when I said you had a known sequence of the genome with you that was all of a length 12 kb. Then I said this is a plant of 12 kb, mapping of which was already known to us. But when we were comparing A with the B and C, we came to know B and C are the mutants because in that 12 kb fragment, certain bands were lost and certain new bands appeared. So this is the reason here mapping is no more possible just on the basis of the phenotypic features. But here is the case where mapping could be done and you could locate different traits onto the chromosome, right? Here, screening at seedling stage for economic traits. I mean to say, here you were conducting no yield preliminary trials and all. And why you were going for those trials? Because you wanted to know whether at the end it is going to produce the say desired amount of yield or not. And if you are focusing on the yield, that itself clarifies like you are focusing yourself on the economic aspect. More will be the yield, more will be the improvement of economy, right? Here you cannot screen at the seedling stage because you need to wait here for at least 7 years if it is 12, 10 to 15 years to conduct that preliminary yield trials. You have to wait for at least 5, 6 to 7 years, right? But here screening, like whatever we are modifying, whatever we are going for, is it going to be is it going to improve your economic structure or not? That itself can be screened at the seedling stage only. So this was all about your molecular markers and advantages and uses of molecular markers. Somewhere I have often made you all familiar with a certain say disadvantages like which are cost related and all. So this was all from my side. Thank you and stay home and stay blessed.